In our paper, a predictive model for selective targeting of the Warburg effect through GAPDH inhibition with a natural product, Liberté explored properties of the cellular metabolic network that can change enzyme activity and glucose metabolism to selectively target cancer cells. Using metabolic control analysis, they found a way to disrupt metabolic pathways important in tumor growth without affecting normal cells much. They thereby provide the basis for a novel therapy that selectively kills cancer cells based on their metabolism. Glycolysis is covered in Module 3 and is a part of the process through which organisms convert glucose into energy. In this experiment, the substances involved in glycolysis are used to measure the response of cells by studying their glucose uptake and lactate excretion. Glycolysis intermediates are also used to analyze this process. If the levels of an intermediate increase, that means the reactions in which they are involved in are blocked. Glycolysis is the metabolic pathway that converts glucose to pyruvic acid by a series of 10 enzyme-driven reactions that involve splitting the 6-carbon chain of glucose into two 3-carbon chains of pyruvate. An enzyme is a substance that does not participate in a chemical reaction, but instead acts as a catalyst and allows the reaction to happen at a rate much faster. The steps of glycolysis need enzymes to happen at a rate fast enough to sustain life, and one of these enzymes is GAPDH. The Warburg effect is a phenomenon in which most cancer cells predominantly produce energy by a high rate of glycolysis followed by lactic acid fermentation, instead of a low rate of glycolysis followed by mitochondrial oxidative phosphorylation, as in most normal cells. Rapidly growing cancer cells typically have glycolytic rates up to 200 times higher than those of their normal tissues of origin, and the paper's authors researched whether this difference in metabolism could be used to target cancer cells. One major difference in the cancer cell's metabolism that could become a therapeutic response was the role of GAPDH, the rate-limiting enzyme that contributes to the breakdown of glucose in both normal and cancerous cells. Their research showed that GAPDH is more important an enzyme in cancer cells in regulating energy production. They then established a natural product produced by fungi, koenigic acid, or Ka, to be a selective inhibitor of GAPDH, which inhibits GAPDH's substrate and prevents cancer cells from making energy. Targeting GAPDH with Ka therapeutic properties on cancer cells while having minimal adverse effects on normal cells. This targeted cancer therapy is therefore capable of differentiating normal cells from cancer cells based on their genetics and exploits their differences during treatment. The team used metabolic control analysis to find out how metabolism processes were different in the Warburg effect. They found that only GAPDH showed more control over metabolism than the Warburg effect in regular cells. To test this, they inhibited the action of GAPDH using Ka in studies on mice tumors. Ka showed a dynamic effect on glycolysis. The metabolites before GAPDH accumulated within hours, while those downstream depleted in minutes, but then recovered. This suggests that metabolism is limited when GAPDH is inhibited by Ka, making it a possible way to slow down tumor growth. The fungus that makes Ka manufactures GAPDH resistant to Ka inhibition, so that only its attackers are affected by the inhibitor. When Ka-resistant GAPDH gene was replicated in mice cells and Ka given to them again, they showed no change in metabolism, suggesting that Ka affects the activity of GAPDH. In the cancer cells which were not resistant to Ka, tumor metabolism was affected. Tumors' growth, proliferation, and morphology were all highly altered in the Ka-sensitive tumor. However, levels of other biological processes in mice were unaffected, which suggests that Ka is tolerable to mice and slows cancer growth. These two graphs compare tumor growth in cancer cells with the Ka-resistant enzymes and the regular enzymes in the graph on the left over time. The enzyme GAPDH was active because it was modified to resist Ka. However, in the graph on the right, GAPDH was inhibited by Ka, as in human cancers. The growth in these tumors decreased over time and their size was reduced. This is because metabolism in these cells was lowered in the presence of Ka, unlike cells on the left, in which metabolism was unaffected by Ka. This shows that Ka does inhibit metabolism in tumor cells, and there was no indication of toxicity in other tissues. Hence, Ka is bioactive in cancer cells, but spares normal cells. The research in this paper showed that targeted cancer therapy is capable of differentiating normal cells from cancer cells based on their metabolism. Since the metabolic network in cancer cells is regulated by GAPDH, which is inhibited by Ka in the Warburg effect. In the future, Ka can be used as a way to slow down cancer growth by inhibiting metabolism in cancer cells with limited effect on normal cells.